Hi everyone! So today we're going to learn how to annotate microalgae to fill out the data entry form for the microbe directory. This is the data entry form that we're going to fill out today. Okay, first things first, I highly recommend reading this Coda doc because it contains a lot of great information on things you will need to annotate and just provides an overall idea of what your microbe is, which is good to have when annotating. Okay, so today I'm going to be annotating the microalgae Chlorella vulgaris. So what I'm going to do is copy paste the name exactly as it appears. It's going to appear in your email. Here it appears on a slide. So I'm going to copy and paste it exactly into the scientific name section in the data entry form. Okay, so let's get annotating. So my initial step as a volunteer is to head over to the NCBI Taxonomy browser. So I've searched it up in Google. It's right here, Taxonomy browser. Awesome. So in this search area, this is where I'm going to copy and paste my microbe. Let's go. Okay. And I'm going to click on the species level because this is where we're annotating at the species level. So the NCBI taxonomy browser, it provides great insight about the taxonomy of the microbe. It may also provide, depending on your microbe, an external resource link. So these are external links that they have provided for us that we can go and head over to just to find more information about the microbe. Something I do want to highlight is that these links are also present in our useful links section in our Coda doc. So you'll find a lot of these links are right here and the databases that are listed under the NCBI taxonomy browser. So yeah, okay, awesome. So let's check out the taxonomy or the lineage. Okay, so for my microbe, I see it says the phylum Chlorophyta, right? So this is essentially green algae, specifying green algae. So I'm going to, it's a eukaryote also, so just to make sure. So it's a eukaryote and it's specifying green algae. So I'm leaning towards microalgae. And even in the email it states it's a microalgae. So eukaryote, algae, macroalgae, we will figure that out. As of right now, I'm going to annotate microalgae. Awesome. Okay. So let's start gathering some information on our microbe. Okay. My favorite website to initially start off when doing research on or annotating a microalgae is AlgaeBase right here. What's great about NCBI taxonomy browser is that when you click right here, it takes you directly onto the page that you need to be reading. As you can see right here, AlgaeBase is also present in our useful links. So definitely check that out. So I'm going to click on that, Chlorella vulgaris. It's going to load. Hopefully load soon. Awesome. So it's loaded. It provides photos and other information that you can actually use, which is great. So I actually see that over here in the general environment, it states freshwater and terrestrial species. Okay, I'm going to keep that in mind because we're going to need this information to just create an overall idea of our microbe. So I'm going to keep this in mind. I also see the locality. It talks about a pond in the Netherlands, which is probably going to be useful. So I'm going to keep this open for later citation and later reference. I kind of know now that it's a freshwater species, and it also says useful notes, potential wastewater treatment, suitable pioneer organism for soil restoration. Ooh, that's interesting. Okay, so I'm going to keep this in mind. I kind of have an idea of what my microbe is. So let's try to head over to another database that I can use. Okay. So it's great that it's in order. My second database that I usually go to is the Encyclopedia of Life. It can also be found right here next. It's shortened for EOL, which is Encyclopedia of Life. Yeah, so I'm going to go ahead and click it. It's going to take me over to the page. Awesome. Okay. So what's great about this database is it actually shows the interactions of your microbe. So like for this microbe, it has a host that's eaten by this organism. But it's also, what's amazing is that it can also provide you with the habitat and sometimes the metabolism. So what I'm going to do is also keep this for later reference. I kind of have an idea that the habitat is related to freshwater. So what I'm going to try to do is try to find evidence or even other databases that provide maybe a little bit more precise location to where this microbe might be found. And yeah, so let's try to find more information. Okay. So the next thing I kind of want to do is actually possibly do some Google scholaring. Let's call it Google scholaring. Before we start, using Google Scholar, I just want to go over a few things that, are going to help you with your experience. So what I'm going to do is go ahead and search up the growth. Okay. So something that's important that you should do is actually read through the titles, but also possibly read the subscript down here that actually tells you, gives you a little bit more insight and just lets you know if that article is relevant to you or not. Now we know how to try to find articles that relate to your microorganism. So what I'm going to do is actually show you how I read through articles. So I'm going to go ahead and click on the article. Oops, this one does not have the full PDF, so I'm going to just click on the PDF form. So that's popped up. Okay, cool. So the first step that you should be taking when reading an article is actually read through the abstract. Just to see if this article is still relevant to you. It might seem relevant on Google Scholar just because it was picking up keywords, but now you're going to read through the abstract and make sure it is actually relevant to you. Another thing that's super helpful to do is actually use the find command. So, and I have actually put in the second half of my microalgae's name, just because sometimes they abbreviate, the first half. So, Chlorella vulgaris. This trick is mostly used for the main introduction and like the other more densely packed parts of the article, just because it helps highlight where your microorganism is being mentioned. So this is a good tool to utilize. However, you also want to, if you want to find more information, I highly suggest you try putting in the first half of your microorganism, because it also shows other places your microorganism has been highlighted. And so you can read through this. If you're looking specifically for, so I was looking specifically for growth, I'm going to put in growth, and it's going to highlight, okay, microalgae growth, various growth conditions, exponential growth. So you can read through these things. And let's say in the article I was looking for maybe biofilm, and I see that there's no mention of biofilm. So that's something you can also use to see if it has any information about something specific that you're looking for. So even I'm going to look in for colonies. No information on colonies. So you can use these tools to navigate through your text, just to make it a little bit easier on just annotating. So another thing that I actually do is that if my organism pops up through the find command on a specific paragraph, I'm going to make sure I read that specific paragraph with greater scrutiny or even like just greater attention, just because it might have more information about the microorganism. So make sure to use these tools when you're annotating because they're super helpful and they're just going to make your life way easier because some of the articles can be very lengthy and it can have information that might not give you any insight on what you're annotating towards. So, yeah. Okay, so I'm going to copy and paste. Oh, right there. I'm going to copy and paste it. Okay, I see a lot of great literature that has popped up, um, which is great for this microbe, but you'll find in your case sometime you might come over a microbe that uh, may not have as much information. It might be unknown, so there's going to be a lot of missing information. You're going to have to do a lot of great digging. So that's that. So what I'm actually interested in is it's talking about autotrophic, heterotrophic, and mixotrophic growth conditions, which is something I am interested in. So I'm going to go ahead and click on this one, and I'm going to give it a quick read and giving it a thorough read to find information that might be useful in annotation for my microbe. Okay, so after giving this article a quick and thorough read, I was able to confirm three things. So, three things I was able to confirm was that our microbe is in fact a microalgae. It is not a diatom. Its microbiome, we still have to do a little bit more digging. But we know it involves water, it involves fresh water, and there was some talk about a pond, so I'm going to highlight that. I'm going to go back and just check later. So the second thing I was able to confirm was that our microbe has the ability to do autotrophic, heterotrophic, and mixotrophic metabolism. The third thing that I was able to confirm was that our microbe can grow colonially. So what I want to do now is actually find more information about the isolation or the microbiome of our microalgae. So what I'm going to do is go to another database that I actually find super helpful because it provides a lot of information about where your microbe is isolated from. It's called the Global Catalog of Microorganisms, and it's super helpful, and it just provides insight in where the microbe is found. Right here is this column that tells you where it's isolated from. So what I was able to confirm was that it is found in pond, but it's also found in urban environments. So this is going to be super important. It's also found on sediment, more specifically freshwater shore. Okay, cool. So we've got so many things covered. Now we're going to move on to some of the more specific questions that are present, so to speak. So we're going to start looking for some pigments, mobility, biofilm, if it's an extremophile, it's reproduction, of course, with spores or not, and then symbiosis, pathogen, content, and resistance to UV. Okay, so what I'm going to do is a little bit more of Google scholaring. So I'm going to go ahead and so what I'm going to do is let's figure out pigments. Okay. Interesting. So I want to look into the protein, fatty acid, and pigment content, and try to see what we can find. So that's interesting. I'm gonna look through anything else that I want to talk about. Oh, so they're talking about extractions of lipids and pigments from Chlorella vulgaris. So I'm going to check out these two articles real quick, give them a quick read. So after giving both of the articles a quick read, I was able to find some more information. So some of the information I actually found was that it does produce pigments, and those pigments are chlorophyll A, C, and beta carotenoids. My apologies. Yeah. So those were the pigments that were found and were extracted. Also, other things that were interesting was content production. So there were content production, and there were proteins, lipids produced that were used, or my apologies, were extracted from the microalgae. So those are some things that I was able to annotate. So I'm going to do a little bit more Google scholaring to find if it's an extremophile, biofilm, mobility, and spores and stuff. So let me go ahead and actually so let's take a look at what we can find. So I see there's a lot of talk about a biofilm reactor, or there's use of biofilm assistance. So I'm not sure what this is specifying, but we can take a read and just see what's there. I'm going to check out some other ones.
Six and a half hours later.
So after reading a few articles off of the Google Scholar results that I've got, I've come to the conclusion that our microalgae does produce biofilm. And second conclusion that has come up in an article is that it is not an extremophile. So I'm going to go annotate that, and I'm going to cite those articles. I'm going to actually cite those articles later. So I'm going to keep the tabs open. Awesome. So I want to look into more of the mobility and the reproduction. So let me do a little bit more Google scholaring. So what I'm looking for is mobility. So I want to look for if our microalgae is mobile or not. Motile, excuse me. Yeah, so let's see. Okay. There isn't much talk about the mobility. They're talking mobility in different terms. So, okay. So what I'm going to do is kind of read around a little bit, see if I find anything. So after trying to find more and more resources to see if we talk about mobility anywhere, I was not able to find information about mobility. So what I'm going to do is actually annotate unknown for mobility, and I'm going to move on to reproduction. So I'm right here, reproduction. I like an article that I see, so I'm going to go ahead and read it. Um, Give it a quick read. What might be better is actually getting this specific article. So I'm just going to copy and paste this specific article. Aha. So I looked over this article, did a quick reading, and I actually was able to confirm a few things. So it turns out that the microalgae has the ability to do asexual reproduction. I wasn't able to find much evidence about sexual reproduction, so I'm going to annotate asexual reproduction. I also found that it spores, so I'm going to annotate yes. But what was really cool was that I actually found a little bit more information on some isolation. So in the article, it actually stated that the microalgae can also be found in wastewater. So I'm going to go ahead and annotate that. This is great because it actually shows you that when you go through different articles, you'll actually find more information that you can actually annotate. So that was awesome. So what I'm going to do now is actually try to find more information about the pathogen. Okay, so let's see. I'm going to take a quick read, try to find more information. So I went through a bunch of the Google Scholar links and I read through some of the articles, and I was able to confirm that our microalgae is not a pathogen. If you go back to Encyclopedia of Life, um, excuse me, you'll see that it doesn't really state that it has it is a pathogen to another organism. Rather, it shows that it has a host and it's eaten. So, just more validation that we got. So the last thing I actually want to look into is the UV resistant. So I actually found some information from an article uh, that I used in the annotation, and this was the article where it talks about effects of UVC radiation on Chlorella vulgaris. So I'm going to go ahead and give it another quick read, see if it talks about any resistance to UV radiation. There is some information in this article about the microalgae's resilience to UV, but it doesn't really give much of a confirmation. So I'm actually going to go through Google Scholar again and try to find more details on just, you know. Okay, so I have found some information, and in the conclusion, it actually stated that it is UV resistant, and it's UV resistant to A and B. There is not a lot of information of C, but it has a level of resilience to C, but it didn't say resistant to C. So I'm not going to annotate that. And yeah, so we're pretty much done with the whole form. So what I'm going to do actually now is open up the zbib.org, and I'm actually going to start to annotate, to cite my information. So I actually want to start off by citing the Encyclopedia of Life. I'm just going to go ahead. It's going to create a great citation that I can actually just copy and paste it right into here. Okay, cool. So now what else I want to cite is actually the global catalog of microorganisms. So I'm going to go ahead and cite that also. And you just do this. You continually do this to all the tabs that you've opened and actually used for information. So, you can go ahead and go to one of your articles, just copy the link on top of the article, and you can just go back to zbib and boom, there you go. And you just copy this and you go ahead and paste it right here. This is a super easy process. There might be some chances where you might have to manually annotate. So you might want to be ready for that. So sometimes like your link might not work because it's a PDF. So for example, this is a PDF, right? So I'm going to go ahead to just copy the title. You're going to go to zbib. You're just going to put that in. So here you see that the option popped up. It talks about, the title that you put in and states that it's a journal article. So you're going to click it and it's going to produce a citation for you. And you just copy and paste it right here. So yeah. And so what you could do here is just press NA. This is not required. I'm most of the time just put NA and enter your name. So your name. I'm just gonna write test. Awesome. So you have completed your form, and now all you have to do is press submit. And it's going to give you this, and it's automatically going to submit.